All right, class, today we are going to cover the cellular process known as mitosis. Okay, so first off, what is mitosis? And the answer can actually be found in this picture right here. Okay, this right here, you've got a nucleus right here, some cell organelles. So this is obviously a cell, and what's it doing? It's doing math. What kind of math? All right, it's doing division. All right, so that's basically your answer to what mitosis is. It's cell division. All right, start off with one cell, and you end up with two identical cells. Okay, now you might think, why would an organism do this? All right, first off, if you were a single-celled organism, all right, like bacteria or protists, okay, mitosis results in asexual reproduction, and that's basically how single-celled organisms reproduce, all right, through this process right here. Okay, now what if you were a multicellular organism such as yourselves? When do you think you would have to go through cell division? When would you add more cells to your multicellular body? All right, and the answer to that is basically, think about the size you were when you were a baby. Are you guys the same size now? Okay, now to increase that much mass, all right, obviously you have to increase the amount of cells in your multicellular body. All right, so growth and development would be one reason why multicellular organisms go through mitosis. Okay, now what would be another reason? All right, what do you think happens when you get injured? You get a cut. What happens? Okay, your body has to repair itself. All right, so that is the other reason why a multicellular organism would have to go through mitosis. Growth and, of course, repair. Okay, anytime a multicellular organism needs to add more cells to its multicellular body. Okay, do you understand that it's a process? All right, you do start off with one and then you end up with two. All right. I don't know if you guys are old enough to watch Twilight, but there was a meme going on where it's a bad love story. Okay, so here you've got mitosis again, asexual reproduction, and it's still a better love story than Twilight. Okay, what happens to the other cell organelles? Okay, they're there. It's just that for the in order to learn about mitosis, we do kind of ignore them. All right, we do concentrate on what's important. All right, and what's important is the DNA and the organelles that move the DNA. All right. Now, during cell division, during mitosis, all right, your centrosomes or your centrioles, all right, they will be the central areas as to where your DNA will be moved to. All right, and your cell will create structures made of protein to actually move this DNA to that area. Okay. Now the other organelle we're interested in are your chromosomes. Okay. During the cellular process mitosis, your chromosomes, all right, are your DNA. Okay, your DNA actually folds itself up into the shape of a bar or an X. Okay, and this folded up packaged DNA, either a bar or an X, is known as a chromosome. Okay, so DNA, once it's packaged, folded neatly into a bar or an X, is called a chromosome. And these are the two structures we will be concentrating on during this lesson on the cellular process mitosis. Okay, first off, interphase. What is interphase? All right. Do you understand that a cell also has its own life cycle? it doesn't just always continuously go through mitosis over and over again. All right. So the cell's life cycle outside of mitosis is known as interphase. All right. And there are three parts to interphase. Okay. You've got G1. Okay. Where a cell grows and develops. Okay. There's also an offshoot called G0. And during G0, a cell does zero growth. And this is where a cell will do its job. All right. If this is a unicellular organism, then at G0, it would live out its life. Okay, this would be its life cycle right here. Okay, now if a cell has to go through mitosis, okay, it leaves G0, goes back to G1, and then in turn G1 will move on to S cycle. And during S cycle, the DNA doubles itself. All right, and then once S cycle is finished, it will move on to G2. G2 double checks everything, make sure that all the DNA has been doubled and replicated. Okay, so G2 double checks. Okay, and of course it goes into the fourth cycle of a cell's life cycle, your M cycle or mitosis cycle. All right, so interphase is the part of the cell's life cycle outside of M mitosis cycle. All right. Okay, so here is the picture representation of what I just stated. Okay, so first let's start right here. Okay, a single cell. Okay, now this single cell, for example, just came out of mitosis. It's a baby. It's small. It has to do some growing. All right. Once it's done some growing in a G1 cycle. Okay, G1 stands for growth or gap one. Okay, so it's either growing or it's a gap in which it's staying still. Okay, during this part of the cell's life cycle, it may go into G0. Okay, during G0, it does zero growth. Okay, growth or gap zero. So in this case, if it's a cell in a multicellular organism, it will do its specific function or job. Okay, if it's a single cell unicellular organism, this would be where it would live out its life. Okay, its life cycle right here. Now, if it has to reproduce, if it has to repair, if it's a multicellular organism that has to repair or grow, right, this cell will go back to G1. Okay, it will then enter S or synthesis cycle, and during this time, the DNA will be doubled. All right, do you understand that the doubled DNA has to be identical on both sides, because at the end, you end up with two identical cells. All right, from there, once it's done through going through S cycle, all right, your chromosomes will look like X's, all right, because it has doubled. Okay, if it was single, it would be in the shape of a bar. Okay, you guys see how it's like in the shape of a bar here, and then once it has doubled, it looks like an X. Okay, it's because one part of it, all right, will kind of synthesize or kind of replicate the other, right, and then the other will actually be identical. So when you have a chromosome, an X-shaped chromosome, both sides are identical. All right. Okay, from here, all right, it goes into a gap or growth 2 cycle of the cell's life cycle. Double checks the DNA, make sure it's good, and then from there it will go through M cycle. Okay, and because M cycle is complicated, we will go through each of the phases of M cycle, and that is your lesson on mitosis. Okay, so here's another representation picture. Okay, single cell. All right, go through your different phases. All right, prophase, metaphase, anaphase, telophase. All right, and this one just concentrates on M or mitosis cycle. Okay, and we're going to learn about them separately next. Okay, since I can't do this with you guys, move on. Okay, first prophase. Okay, The main distinguishing characteristic is always what happens to the DNA. Okay, so in this case, because they've been packaged nicely, neatly in the shape of a bar or an X, they are called chromosomes. Okay, so at the start of prophase, mitosis cycle, you have to have your X-shaped double chromosomes. Okay, because your single chromosomes, all right, your DNA during synthesis cycle start to double. All right, so at the beginning of M mitosis cycle, prophase, you have to have X-shaped double chromosomes. Okay, that's the most important characteristic. Okay, now your centrioles might be at opposite ends, and they'll start pushing and moving the chromosomes to the middle of the cell. Okay, the nuclear membrane will break down. All right, the membrane, or the little film of lipids that wraps up all your DNA and keeps it nice and safe in the center of the cell, breaks down. All right. Okay, and here's a little picture representation of what prophase would look like. Okay, do you notice the X's are the same color? Okay, because this side is identical with this side. Okay, now this one's pink, this one's green. Therefore, you know that this and this is not identical. All right, if they're the same color, they're identical. All right. Here's an actual fish cell going through a prophase. Okay, you guys can see the condensed chromosomes in the center. Okay, metaphase. Okay, your X-shaped double chromosomes are now lined up in the center of the cell. Okay, and every chromosome is connected to what's called a spindle fiber. All right, your spindle fiber is created by these centrioles. Okay, these are proteins, and these are cell organelles that create more and more proteins. So in this case, they kind of use these protein strands called microtubules to push the X-shaped double chromosomes in the center. Okay, and here you go. Here's the centrioles, the protein. It spun a protein strand that pushed the chromosomes into the center. Okay, same thing with the other side. They work together in conjunction to push and pull and make sure the X-shaped double chromosome is at the center of the cell. All right. Okay, here's a actual cell in metaphase. All right, in the center, you can't see the centrioles. All right, very very small. Okay. Okay, your next phase is called anaphase. All right, so the thing is, metaphase is very short. All right, prophase is kind of long. Metaphase is kind of short. Anaphase is pretty long as well. Okay, so as soon as the X-shaped double chromosomes have been ripped apart into single chromosomes, all right, you guys get it? Single chromosome, double chromosome. Okay, once they've been ripped apart into single chromosomes, all right, that's the beginning of anaphase. Okay, now during this point, all right, the centrioles, what actually rips them apart are the centrioles. Okay, so the centrioles and the spindle fibers start pooling from both ends, and then they kind of just rip them apart. All right, and then as soon as they're ripped apart, it's anaphase. So main characteristic, always about the DNA, double chromosomes are ripped in half by the pooling of the microtubule fibers into single chromosomes. All right. Now, you may also call these chromatids. All right, single chromosomes sometimes can be called chromatids. Okay, but they will start moving toward the opposite ends of the cell. Here's a little picture that I created of your anaphase. Okay, the X-shaped chromosome has been ripped apart into the single bar-shaped single chromosomes. Okay, and it's these centrioles right here that start pulling in this spindle fiber here and here, and of course, once they do it at the same time, they kind of just rip it apart. All right. Here's a picture representation of a fish cell going through anaphase. Okay, these are your single chromosomes moving toward opposite ends of this still yet single cell. Okay, now telophase, and cytokinesis. All right, telophase is what the phase is called. Cytokinesis is not part of a phase. It's just the process of when the cytoplasm of the cell splits, and that's what cytokinesis is. Okay, cytokinesis happens at the end of telophase. Okay, now again, the main characteristic is what happens to the DNA. All right, now two new nuclear membranes will begin to form around the single chromosomes at the opposite ends of the cell. Okay, centrioles will start to disintegrate. All right, break down all of the spindle fibers. Okay, they will rest next to the newly formed nucleus, and then cytokinesis happens. All right, once cytokinesis happens, the cell has been split in two, and that signifies the end of mitosis cycle, and you now end up with two identical cells. All right. Here's the picture representation before cytokinesis. All right. Do you know that they are identical in terms of DNA? Okay, this cell right here has one bar green, one bar pink. Same thing, one bar green, one bar pink. All right, so the DNA is identical. Cytoplasm will split, and then you end up with two identical cells. All right, and here is a picture representation. All right, you see the nuclear membrane forming, creating this round structure of the DNA, and this is again before cytokinesis. All right, and that's all for mitosis. Okay, once a cell comes out of mitosis cycle, it will then enter back into G1 cycle, most likely G0 cycle. All right, where a cell does its job. Okay, or live out its life if it's a unicellular organism. And that's all I have for you guys today.